All right, let's jump straight into this. So welcome to From Pipelines to Interactive Analysis, closing the loop with Seqera Studios. So my name's Rob Newman. I'm a product manager lead at Seqera. I'm responsible for everything related to the platform. And today I'm really excited to be joined by Edmund Miller, my colleague who's a bioinformatics engineer at Seqera, and also Jon Manning, who's a senior bioinformatics engineer at Seqera. So before we begin, we're just going to do a little bit of housekeeping. Please, all questions should be submitted in the Q&A box, and we'll go over those questions and answer as many as possible in a time permitting at the end. And then also, please participate in the closing poll before the live Q&A towards the end of the presentation. And lastly, we'll be sharing the webinar recording by email in a day or two. Okay, to get us started, what we're going to do is I'm going to give a brief overview of Seqera, talking about our product suite and our mission. And then I'm going to jump into discussing the challenges of the modern data lifecycle. After that, I'm going to talk about Seqera Studios for interactive analysis. And this is our flagship product that allows interactive analysis within the platform. And then I'm going to hand it over to Jon and Edmund, who are actually going to give us a live demo of Seqera Studios. And then once they wrap up, we'll have some time for Q&A with you all. Okay, quick overview of Seqera. So a little bit about us. Nextflow is a really important part of the Nextflow story. Nextflow, as most of you know, is an open source workflow manager, and it was created at the Center for Genomic Regulation in Barcelona about 10 years ago. The creators of Nextflow are actually the founders of Seqera, and the first ever Nextflow commit was over 10 years ago, and it was by Seqera's CTO and co-founder, Paolo. So Seqera is really the team behind Nextflow, and many of us contribute heavily to its ongoing development. Nextflow alone is very powerful for writing and running analysis pipelines, but there's so much more that research organizations need to accomplish to scale their research. They need to be able to manage complex infrastructure. Their analyses need to be reproducible. They need to collaborate on the development of pipelines and securely share the workflow results with their colleagues. Hence why Seqera was founded, and to create and offer our commercial platform, Seqera Platform, to meet all of these needs. Additionally, and in parallel, the independent NF Core open source community was established in 2017 to define pipeline development best practices. And although we work closely with this community, they're not affiliated with Seqera, the company. So fast forward to today, we see over 2 million Nextflow executions per month. So it's really become the gold standard for bioinformatics workflows. And Seqera's commercial platform has over 130 customers, many of the top pharma, lots of mid-sized biotechs, and large institutions like the CDC. And you can see some of the logos of some of our customers along the bottom of the slide there. So at Seqera, our mission is to advance science for everyone through software, to provide scientists with the tools, the resources, the world-class software to move their own scientific discoveries forward. We aim to do this with 3 pillars: build, discover, and scale. The first is developing and contributing to open source software so scientists can easily build the analysis tools they need. That, of course, includes Nextflow for writing and running scalable, reproducible pipelines, but also MultiQC for aggregating results and reporting, and Wave, Seqera's container provisioning service for assembling, augmenting, and deploying containerized images. The second pillar is nurturing an open science community. So we not only want the tools to exist, we also want to share the world-class resources and enable scientists to discover and learn from each other. This is through things like Seqera Pipelines, where you can find off-the-shelf ready-to-use pipelines, or finding Seqerans as very active members in our online communities like Nextflow, NF Core, and Seqera's own online community forum. And lastly, we want to enable scientists to really scale their research and their analysis through Seqera's software offerings. So we have the Seqera platform for deploying, launching and monitoring Nextflow pipelines at scale. We have our add-on product Studios, which we're going to be talking about today for launching interactive analysis notebooks. And we also offer Fusion for reducing costs and wall time by giving compute resources direct access to data and object storage. Again, to reiterate, the goal with all of these pillars is to ensure scientists have the foundational tools that they need, that they can learn from and contribute back to the scientific community, and that they can scale their research in a way that's easy and, most importantly, reproducible. Okay, going through the challenges of the modern data lifecycle. Okay, there's 5 main challenges that we've noticed with our customers around the data lifecycle and analyses. First one is data transfer. Transferring the typically large data sets commonly used in bioinformatics from cloud storage to your local hosts for direct analysis wastes time and it costs money in egress charges. For example, Amazon S3, after you've gone through your first 100 gigabytes of egress per month, it's 9 cents per gigabyte for the first 10 terabytes that you download. Also, transferring data between regions also incurs charges for both the storage and the destination region, with egress charges typically around 9 cents per gigabyte as well. The second is silos. Commonly, data sources across R&D bioinformatics and analysis teams are distributed across regions, making collaboration difficult. Or worse, and we've actually seen this, is data is local to just someone's personal computer. Secure sharing is caring. Thirdly, compatibility. Analysis tools and libraries are often installed in an ad hoc manner per analysis session, and they're not necessarily pinned to specific versions, or there's no institutional level control or verified libraries or version-controlled collections of tools. This makes creating reproducible analysis environments extremely difficult and creates confusion and inconsistent results between different analysis sessions across teams. Fourthly, compliance. Different user types require different levels of access. Some users need only read-only permissions. Some users need to only connect to existing sessions and run analyses, but they don't actually need to manage the analysis environments themselves. And some users need to manage both the type of analysis environment and the overarching compute provisioning. For example, does an analysis need GPUs to efficiently run? If so, who's responsible for making those compute environments, and where are they taking those actions? Doing this at scale is hard. We've seen multiple customers spin up their own fleet of EC2 instances just for interactive analysis, which results in needing to manage infrastructure and permissions around that fleet. The fifth one is provisioning. There's nothing more frustrating if you've done scientific analysis than running out of memory or disk during an interactive analysis lifecycle. To compensate for this, administrators will typically intentionally over-provision to ensure you have an uninterrupted analysis session. This results in unused compute resources that you're often paying for. Additionally, if you're managing fleets of EC2 instances, you need to ensure that they have lifecycle management, which is just another item for your infrastructure team to manage. Provisioning may also require layers of approval from your institution, delaying the time that you actually get to do your work. Okay, so let's talk about how Seqera Studios and our interactive analysis suite solves this. So there are 5 key ways that Studios addresses these problems. The first one is seamless integration with your existing infrastructure. So with Studios, you can effortlessly co-locate your data, compute and analysis tools within your existing cloud infrastructure while ensuring compatibility and data integrity without disruption. Secondly, optimized resource management and cost efficiency. With Studios, you minimize the data transfer and storage costs with on-demand compute environments that scale as needed in the same region as your data storage, and that's really key. It's adjacent to where you do your object storage. And also you have centralized visibility into usage and expenses, and you only pay for what you use. Thirdly, enhance security and compliance at scale. You need to protect your sensitive data with secure authentication and role-based access control and automated notebook security, while maintaining full audit trails and checkpointing for traceability. Our Seqera provided container templates ship with scan results and build reports accessible to you. They're actually built and managed by our Wave container provisioning service. And by default, Wave does security scans and creates inspection reports. And those are available for you whenever you need them. In terms of collaboration and secure data sharing, Studios removes research silos by enabling secure real-time collaboration across teams with the ability to do shared interactive sessions and also one-click access to your reports and analyses. Alternatively, you can also run your analysis sessions in private mode if you want to. This is a relatively new feature that we've rolled out and I'll be discussing later on. Finally, velocity. With using Fusion under the hood, Studios really lowers the time to insights. And Fusion also offers a true POSIX interface. So typically what we're seeing is 2 to 3 times velocity and throughput with our customers that are already using Studios. And this is due to Fusion under the hood. Okay, let's talk about some of the features and go through them by category. I've broken these down into like 6 different sections. So the first one is collaboration. So within Studios, we have role-based access control. Some roles have view only, so they cannot actually connect to the running session. Different roles have the ability to connect to a running session. And then we have additional, more administrative roles that actually allow you to manage those sessions. So starting, stopping them, adding them, removing them, reconfiguring them. Additionally, and as I mentioned in the last slide, we also have the ability to not only create a shared collaborative environment, but also you can give, or you can define your studio as having a private option. This means that only the creator of the session can actually connect to it and do the analysis. So it's like true privacy. Also, with many of our environments that we provide, you can do real-time multi-user collaboration. And within the environment, you will see different avatars with different colors for the different users who are actually in those sessions. Finally, in terms of collaboration, you can also take an existing studio session and you can fork it. And we're calling this add as new, which is you take an existing checkpoint from a session and then you can create a brand new session. So if you want to do some sort of exotic analysis and you don't want to impact how your team is using the main studio session, you can do that in your own one. And then if you choose, fold those results back in. Okay, moving on to checkpoints. Within Studios, as a studio is running, or as a session is running, a snapshot of the environment is taken when the session is first started, and then every time it's stopped. Additionally, within the session itself, a snapshot is taken every 5 minutes. And what this allows is a level of auto recovery. So for whatever reason the session crashes or errors out, you do have the ability to go back to the most recent view and be able to access the work that you've done. Additionally, and as I mentioned before, you have the ability to create a new session from any historical checkpoint. So if you have a very long-lived studio session for many months, even years, you can go back to any point in time that that session was stopped and started and take a view of that environment as it existed at that point in time and create a brand new analysis environment. Finally, and this is also a relatively new feature that we've released, is you have the ability to migrate or update between different session templates. So for example, if your version of R has updated from 4.40 to 4.41, we have the ability to do a best effort at migrating the studio session to a different or more recent versioned analysis environment. Okay, environments. So by default, we actually provide 4 templates for you to use out of the box. This is our Studio server, which allows you to do analysis in R. And actually, Jon and Edmund are going to be demonstrating that. We also provide a Jupyter Labs environment. So that's for Python. And then we also provide VS Code. And the VS Code actually does ship with the Nextflow language server already embedded in it. So if you want to do Nextflow development within Studios, you can do that. And then lastly, we also offer Xpro, which is an open source remote desktop software package. So if you want to do specifically like graphical user interface applications, like IGV, for example, you can do that within Xpro. We also offer semantic versioning of those provided templates. So we have our own Connect server that does a lot of the functionality under the hood. And we have semantic versioning of both the library, such as RStudio or Jupyter, and also our version of Connect. As I mentioned earlier, we also do on-demand container security scans and build reports provided by Wave. And then also, you can do environment customization using Conda. So this is really key. You can take our provided template and you can augment that with your own libraries. And the reason for doing this is we don't want to provide a container that's very bloated and heavy and try to guess what analysis tools you need for doing your interactive analysis. So you can drop in either a YAML configuration, like just copy and paste, or you can upload a YAML file. So if you have a version controlled environment that you manage, you can upload that directly into the Studio session and it will accept that, those libraries that you're installing. And additionally, you can pin those libraries to specific versions. We also offer the ability for you to bring your own public container. So if you have an ECR container hosted somewhere, or if you have it on Harbor or somewhere like that, if it's publicly accessible, you can just drop that into a form field and we'll accept that. And we also have instructions on how to build containers so they're compatible with Studios. You can also use your own privately hosted containers if it's using Amazon ECR. In terms of configuration, we have a workspace setting for the session lifespan. So you can choose to have unlimited lifespan, or you can choose as an administrator to say, hey, I only want 4 hours maximum for the lifespan of my studio set for my team's studio sessions. And at that point, shuts down gracefully and takes a snapshot. So again, if you set at the workspace level, your session to be an unlimited amount, when people create their own studio sessions, they can choose to customize the lifespan as well. So they might only want a session to be alive for 2 hours, 4 hours, 6 hours. We also offer different compute environments. They can be CPU empowered, GPU empowered, or hybrid. We support all of that. And also, we support the ability to attach or mount EFS volumes. So for example, if you have your organization utilities, maybe a bunch of libraries or binaries, and they're stored in an EFS volume, you can mount that EFS volume so you don't have to install any additional libraries, and they're always the single canonical location. And as I mentioned around snapshots and environments, your in-session installed libraries can be saved as well. And finally, many of you use the CLI, Seqera CLI, for managing your infrastructure. We also have support now for Studios via the CLI for automation of creating, managing, and removing your studio sessions in the platform. In terms of data, you can mount any cloud storage bucket, and this is using Fusion under the hood. You can choose to have that as read-only or read-write. Public and private buckets are supported, again, with the assumption that you've added your credentials to the platform and you can actually have access to those buckets. And Fusion is a true POSIX compliant file system. So it feels very much like you're in your terminal on your local host. And with managing or with using Fusion under the hood, we're typically seeing 2 times performance improvement for accessing files and reading files. And finally, cost allocation. This is another new feature that we're currently releasing or has been released into preview. We offer the ability to attach resource labels. So that means a key and a value to a studio session. If your compute environment that you're using in the platform already has resource labels attached to it, those are attached to everything related to the studio session by default, but you can choose to edit those and remove them if you have a maintain role or above. And then also what is interesting, if you're a root or an admin user within your, if you have an enterprise install of Studio or the Seqera platform and you have admin privileges, there is an in-platform dashboard where you can go and see which organizations and which workspaces have studio sessions running, and there's a count of all of them. We don't do true cost estimation yet for studios. That's in the roadmap, but this is a proxy for that as it stands right now. And here's an example view. I think in the interests of time, I think I talked longer than I expected. I'm going to kind of move it along to Jon and Edmund. But if you go to the landing page of Studios, and this is enabled in the cloud platform right now, it's been available in enterprise versions since 24.1. You can actually go in and you can see these different features that are in Studios. So here on this screenshot, you can see that we have a couple of sessions that are in private mode, so no one else can access them. You can see the resource labels, which is the third column in. Then you can see the compute environment. Actually, I have a helpful slide for that. You see resource labels. You can see the managed templates, or if a customer has bought their own template, that has a different icon. And then you can see if a session has a lifespan attached to it, you can see the session stop time column. And then also you can see how many users are actually connected to your running session. So in this example here, there's one session that's got 3 users attached to it, and there's one session that has 1 user attached to it. And then also the last column has the role-based access control. So depending on your user in the platform, you will have the ability just to see this view and click in and see the detail view, or you'll have the ability to connect, to start, to stop, to delete. It all depends on your role in the platform. Okay, and with that, I'm going to pass it over to Jon and Edmund, who are going to talk you through a demonstration of Studios in real life. So I'm going to stop sharing.
Thank you, Rob.
And hand it over to you, Jon and Edmund. Thanks.
Thank you. So yeah, Edmund and I are just going to walk through a kind of basic example of how Studios could be used, picking a common use case from the NF Core community. If you've used Platform, this screen might look familiar to you, but otherwise, essentially I have 2 workflow definitions here, one of which is the NF Core RNA-seq workflow, and one of which is the differential abundance workflow, which is designed to work downstream of RNA-seq to produce some exploratory analysis and also some differential analysis of those data. And ahead of this session today, I have run RNA-seq, and I've also run differential abundance twice, in fact, and the reasons for that will become clear as we go through. And yep.
Could you zoom in a bit?
Sure.
On the browser. Thank you.
Okay. So the reason we picked this example is actually because differential abundance produces a Shiny application as one of its outputs. So we're going to use that application to demonstrate how we can use Studios. Before I start, it's also important to highlight the Data Explorer functionality that we have in Platform. This allows us to actually browse the contents of Cloud buckets. And in particular here, I've created a link to a specific path within Cloud bucket that contains the results of these workflows that I'm going to be using today. So I have the results of my RNA-seq workflow, and I have the results of my differential abundance workflow. And I can browse them there as if they were kind of a local file system. So with those things in place, and these actually within these results, there is the Shiny app that I want to use. I'm going to use those to connect up a data studio. So the example that I'm going to pick today is we are going to use our studio, as Rob mentioned, but we're also going to augment that with some custom software. So if I just point at the documentation in Seqera Platform right now, there's some documentation here that shows you how to build custom containers, and they look something like this. So you basically build a Docker file that imports the Connect client, which provides the connectivity between the application and the Seqera Studios. And then there's another line that allows you to supply the port that the application is served on, and kind of the application logic sits in between those 2 things. So in my specific example, my Docker file looks something like this, and don't be scared if you're not used to looking at Docker files. I just want to illustrate the principle that we have the connect client imported at the top here. And then I've just kind of added a bunch of logic in here to provide the software that I'm going to need to do the downstream analysis. And I'm going to use that Docker file to build a custom container, and store that in our public Docker repository, and use that to build my container. And you could use quite a variety of different software tools yourself to build custom containers in the same manner. So going back to the platform page, I'm going to click on Data Studios. And I'm going to click Add Data Studio. By default here, I've got the compute environment that I defined. So when you run either workflows or studios in platform, you have to define a set of compute infrastructure to use, which we call a compute environment. You have to specify the number of CPUs to use. I'm just going to change these. Cool. And then you can mount the data through that Data Explorer functionality that I just described. So here I'm going to select the results for webinar link, and I'm going to click add item and mount it. And that means that when I start the studio, I should have access to all those files. In the general configuration, I'm not going to select any of the pre-built templates. I'm going to use a pre-built container image. And I'm going to use the container URI that I built, which is stored on our public Arbor instance. And the name is just your custom name of your choice. And then you'll notice that at the bottom here, there is a lifespan selected. So if I didn't interact with this, if I didn't interrupt it, this session would time out after 8 hours, basically. And I'm just going to click Add and Start. That will take a few minutes to start because it has to interact with AWS to provide the compute resource. So for that reason, we have some pre-built items in place here. So I'm going to start with one of these. And if I click Connect, then I'm brought into the RStudio interface that was defined in that container. And if I go to the file browser in RStudio, you can see that this data directory has links to that results for webinar link that I defined within the platform, and all those files in there. So I really do have an analysis interface here which has access to all the files as if they were a local file system. But still, we're sitting on the cloud and we're sitting next to the data. So there's been none of those egress fees that Rob alluded to. So I'm just going to check that I should just point out from the interface here that I have run multiple runs of those workflows. I think I may have pointed that out earlier. So I have 2 sets of results from differential abundance, and right now I'm just going to interact with the first set of results. So I'm going to change to that directory in RStudio, and I'm going to start. And this code is produced by the differential abundance workflow. So I'm just going to use that to spin up an application and check that it works. I haven't defined some of the default options, so some of these values are set to null, but the data are still in there. I'm just checking that these things are working. Yes, it seems to be working. The principal components plot is in place. I can change some options. And yeah, it's all good. Okay, so I'm now confident that application is going to work. So I'm going to disconnect from that session. And I'm kind of acting as a bioinformatician here, and my colleague Edmund is going to play the role of almost like a biological laboratory scientist collaborator. And he's going to log on, and we're going to look at the results of that exploratory analysis together, so that we can make some calls about what analysis we might want to do downstream of that. So I'm going to stop sharing now, and Edmund, if you want to share and connect.
Yeah, so just going back to my roots here a little bit. Did an undergrad in biology. So we're just going to play that I am the wet lab scientist who has ran this. John's now set up this app for me. And I'm going to connect. And pull it up here. And I'll just restart the session of the Shiny application. just for the sake of the demo here. And hopefully it'll load. There we go. Okay, so Jon, walk me through this. Show me how to use Shiny in JS.
Yeah, so go to QC Exploratory and look at the PCA.
Okay.
And we have a set of, the data you gave me has some covariates in it. So if you click on color by on the bottom left side of the screen there, you can color by any of the covariates of these data. So let's color by blood, for example, and see which which of these cells are blood and which ones are non-blood. Okay, and the x-axis is the first principal component here, which which is responsible for about half of the variance here. So that's obviously what's causing the most variation in this data. So should we maybe include that as a contrast in our downstream analysis, do you think?
Yeah, I think so. I mean, it also kind of looks like, so you can kind of separate them by the y. If we have 53% here with the x-axis, 27 of the y-axis, So I think that's kind of what we're looking at. But the cell types here that we're looking at are blood and cancer. So we kind of have a crossover of these.
So some of these are blood samples, and some of them, and within that blood status, some of them are cancerous and non-cancerous. So try coloring by cancer status as well.
Okay.
Right, so that's maybe driving the y-axis. Okay, and if you now click under QC Exploratory again and look at the dendrogram.
Okay.
Yeah. And color by cancer status for me. Yeah. Okay, so we can see that we have pairs of cancer and non-cancer within each of the top levels, and I know, for example, that those top levels are blood and non-blood. So, okay, so maybe another contrast we could do is looking at cancer versus non-cancer, but accounting for the effect of blood status in that. Okay.
Yeah, that sounds like a good idea.
Okay, so if you want to share back to me, I'll run the workflow for you.
Okay, so yeah, so you can go back and rerun that analysis for us in this scenario.
Okay, so I'm just going to highlight another feature of platform here, which is data sets. So this actually is the state. This is the data set I probably should have shown you at the beginning. But all of these, we have basically four different types of cell here, and some of them are cancer, non-cancer, blood, non-blood. And I can use this to decide what to do. And Edmund and I's discussion just led us to generate a new set of contrasts we're going to use for downstream analysis. So we're going to have a status like this. So we're going to compare blood to non-blood, and we're going to compare cancer to non-cancer, blocking on the blood status. Okay, so I'm going to launch the workflow again. And I would do that by clicking on the differential abundance workflow here. And I would run that here. And I can actually, within platform, I can then actually select that data set I was just showing you. And I can do other things like control that the front page of that application showed the title properly, for example. So I'm not going to do that because that will take some time. But within platform, we have that second run here, which has those different data in them. So let's just pretend that I run that workflow again with the analysis that my collaborator asked me for, and I'm going to pass back to you now, Edmund, to look at that revised data.
Okay. And so Jon's pinged me or sent me an email, and he's let me know that the results here are now in results 2. So this is our rerun that we just did, and we've kind of pre-baked that solution. So we have results 1 originally here, and then you can see that we've gone into results 2 and have our Shiny NGS app. And those are all connected via Fusion and living in S3. So I've also taken the liberty of relaunching the brand new Shiny app here. You can see we had like null before on that, and then now we have results for Studio Webinar demo on those. And so we can go in, we still have the same PCA plots here. But now the added difference here is the differential expression. And so we want the volcano plots, right, Jon?
yeah, go look in there, and that will show us some of the, and select one of the contrasts on the left here that you're interested in. So let's just select, for example, the blood versus non-blood as a contrast.
Yeah.
Yeah. And on the right, we have the things that are higher in the blood versus non-blood sample. So we can see the immunoglobulins there, which uncontroversially. Maybe if we select the cancer, non-cancer contrast as well.
Yeah.
Got in there.
So prior, this wasn't here. We could only do type, and we can do blood or cancer, right? What was the issue?
So we're just illustrating the iterative approach to the analysis here. So we've been able to go back and forth within studios because this thing is easy to iterate on. So yeah, that's a Hox gene. So we know that Hox genes are developmentally associated, and they're expressed more highly in some cancer cell types. So
yeah, and so that's a transcription factor that's highly expressed in those cancer cells. Nice. And so that's kind of a way that we have passed the data back and forth across the world here in different time zones and collaborated without the hassle of trying to explain how to install Docker and all these other various pieces. So I'm going to pass it back to Rob actually for some slides, and then we'll do a summary as well, and then launch into Q&A.
Awesome. Thanks, Jon. Thanks, Edmund. That was that dashboard is pretty amazing. Actually, Edmund, I think do you want to talk to how the demo actually addressed several of these points?
Yep.
Yeah. So yeah, it was kind of a small example, but hopefully that kind of illustrates the collaborative environment from that. We didn't demonstrate checkpoints exactly on that, but it was the passing between of Jon and I of those, and we could stop and start those studios at any time. For the sake of the demo, we didn't really cover that. We also showed you the custom software environments with Jon's Shiny NGS app and building that, and the Docker file associated with that. Reach out if you're interested in using that in your analyses. And then we also made it possible to provide these rich applications to the analysis for the biologists as well as the bioinformaticians, and collaborate on those and not just pass Excel sheets back and forth. And it kind of quickly enabled that versus trying to set up a server and that living underneath someone's desk and all these other things, or messing around with the cloud and trying to figure out how to get role-based access in that correctly. We didn't demonstrate the cost allocation either, but mainly what we wanted to accomplish here was showing that Seqera Platform is a place to run more than just workflows. You can also do all of your data analysis needs from end to end.
Awesome. Thanks, Edmund. Super informative. Okay, we've now got a couple of poll questions. They'll be displayed on the screen. If you can please take a moment to answer. Okay, so if you want to learn more about Nextflow and Seqera, we've got a couple of resources. We did a very recent blog post about Seqera Studios, specifically talking about 4 real-world use cases for interactive analysis and walking through how Seqera Studios was used to help solve some of these different use cases. And then there's what's really new and hot off the press, the virtual press, I guess, is our LinkedIn newsletter. So you can subscribe to the newsletter and you will get informative information about the latest happenings at Seqera, what we're building, the products that we're rolling out, and any sort of relative interesting blog posts or webinars that are upcoming. Finally, we have the Nextflow Summit in Boston that's happening in May. And I believe, I may get this wrong, but I believe we have early bird pricing still for a little bit longer. I think 15% off. I might get into trouble if I got that wrong, so don't quote me on that. But yeah, that's still open. And if you're interested in talking to both the people that use all our tools and Nextflow, and also the people that make them, there'll be a bunch of people from Seqera, including product managers and scientific development members there, and excited to answer your questions about anything related to either Nextflow or the Seqera platform. Okay, now we're going to go to the Q&A. We've got a couple of minutes. We did overrun a little bit, so I'm going to kind of blast through already submitted ones that we have. The first one was, does Seqera Studio support trusted research environments, secure data environments, and do you have specific documentation on this? This is actually a big push right now for the first half of 2025 is to have trusted research environments. Studios doesn't currently support it, but that is definitely on our roadmap and very high on our radar. Another question kind of related was, do you have US HIPAA compliant environments for patient care purposes? We don't right now. I'm not an expert in HIPAA, so whoever sent that question, I'll be really interested to do a follow-up conversation with you about what that looks like and what we need to do, because that's very much a use case that we have. We are also really pushing towards having GXP compliant environments as well, where anything that you do is logged within the platform. That actually already exists. We have very extensive audit logging capabilities, but both those things are very much on our radar right now. There's a question, is Seqera Studios available for everyone or only commercial? You can use Seqera as a basic or free tier user. You can fire up one studio and have one concurrent running studio. This is to encourage people to try it out and see what it's like and see if it's valuable to you. So you can have one, you can add as many as you want, but you can only have one concurrently running studio. It is a paid product, and I encourage you to reach out to your Seqera account executive to learn about the pricing. When will studios provide full support for Kubernetes, and will there be any additional charges for commercial users already running the Seqera platform on their own cloud infrastructure? Yes, as I mentioned, it is a commercial product. There are additional charges for using studios. In terms of full support for Kubernetes, that is very much on our roadmap as well. We believe that's a relatively straightforward thing to do, and that's definitely for, if not the first half of this year, then definitely by Q3 if we can. There's a lot of feature requests from lots of different people, and I would actually recommend, if you do have feature requests around studios, you can go to feedback.seqera.io, which is our public-facing feedback and feature request system. And you can see the backlog of customer feature requests anonymized. So you don't know which customers requested the feature request. But yeah, you can upvote other people's feature requests, and that obviously affects our roadmap and our prioritization. So please go check that out. Is the Seqera Studios another name for Seqera Platform? No, Seqera Studios is a feature of the Seqera Platform. So it's part of the platform. So the platform is where you can run pipelines and explore data. Studios is an additional feature that's part of Seqera Platform. And how well does it scale with growing user numbers? We've stress tested it with lots of different users in a workspace, but then connected to a running session. I think we've maxed out so far at 10. We haven't really stress tested that beyond the sort of double figures. So super interested in people really stress testing it and seeing how it works for them.
We jump in there,
Okay.
Rob. That's an interesting question. I was thinking about it a different way. I was thinking about the AWS environment and how does that scale? And that was the way that I was going to answer the question, was it hooks into your AWS environment. So as many of these studio environments that you want, you can create concurrent user connected, push it to the limit, and let us know if you run into issues on that.
Totally. Awesome. And with that, that's the last question. We are a little bit over time. Super appreciate everyone's attention, everyone's questions, and special thanks to Jon and Edmund for doing a killer demo. And yeah, looking forward to hearing feedback and you all trying studios. So with that, thanks so much, and hope to see some of you in Boston.